Good afternoon. My name is Howard Jacob, and I want to welcome you to this webinar on the use of whole genome sequencing in clinical practice. I am currently at the Medical College of Wisconsin, and I direct the Human and Molecular Genetics Center. I know we're going to have people coming in and joining on this call as well as the webinar later on. So for those of you that are listening live, I want to encourage you to be able to text your questions in as part of the webinar directions you've got, and then the team will make sure that we're ready to go with that at the end of the seminar. So with that, I'm going to step forward and start off with a little bit of information about what are we doing at the Medical College and where we're going. But before I do that, I want to start off with acknowledging the people that I work with. I'm privileged to be able to tell you about a project that involves a lot of people, and you'll notice on this slide a whole host of people. There's three things I want you to get from this slide. First of all, it's a team effort. Second of all, there's really only 3 physicians that are involved in this entire program. The rest of the people are not clinicians. And the third bit is everybody in the middle are informatics driven. And I think the key point to think about this is that there's a tremendous number of people involved with computers and analysts that's going to be involved as we move more and more down the road of personalized medicine. So again, thanks to my team, and thank you again for joining. All right, so the overview of what I want to discuss today is what is personalized medicine, and I'm going to define what I mean by personalized medicine and why I use the term personalized medicine rather than other terminology that's used to categorize this space. Some people don't think of it as being personalized. They think of it as being precision medicine or something else. But I'm going to be very clear why I call personalized as we go through. I'm going to define the promise and the current opportunity of personalized medicine. We're going to talk some about the ethics and the delivery, a lot around the delivery of personalized medicine, at least how we're practicing it here at the Medical College of Wisconsin. And we're going to use some cases as illustrations as we go through on this. Okay, so personalized medicine, there's a tremendous number of definitions out there. This is my favorite one. It's products and services that leverage the science of genomics and proteomics, either directly or indirectly, and capitalize on the trends towards wellness. I think that's a very key point, wellness, and on consumerism. Again, a very key point, consumerism, to enable tailored approaches to prevention and care. The other part that I think is fascinating about this is that this is a definition that comes from an accounting firm, PricewaterhouseCoopers. And I think that that tells us a little bit about how deep this is going to end up being when accounting firms are starting to think about what is personalized medicine. They're predicting that it's going to have a huge impact on medical care. And I'm going to argue that consumerism is going to be a major driver. And the evidence of that is the direct-to-consumer genetic testing that's already happening and is looking to expand. In fact, we're all going to be hearing about 23andMe as TV commercials as they start launching those nationally this fall. Okay, so personalized medicine, I think this is a slide that illustrates the diversity in the human species. As here we've got Shaq and Patrika. And you can take a look at these two individuals, and we know that they're 99.9% identical, but have huge phenotypic differences in them. And the other part that I think is important is that it's not that our physicians don't practice personal medicine. Of course, when they're working with us, it's personal. But the tools we give them are epidemiological. And I like to use a basic, silly illustration of what I mean by that. So both Shaq and Patrika have a headache. And they go to the medicine cabinet and they read the directions, and it says, adult, take 2 aspirins. Well, we know that they both shouldn't take the same amount of aspirin. He should probably take 4, and she may only take 1. But epidemiologically, we report that on average it's 2 aspirins. And so that's the main point I want to make. One part of personalized medicine is we want to move away from on average. The second part that I think is important is the decisions that they'll each have to make around their own genome. And that'll be a recurring theme that I go through as we talk some more around this. The other part about this, there's a lot of people who say, you know what, personalized medicine and reading genomes is really not that relevant. It's not going to be that important. And I'm going to argue that at a baseline, it's going to be a better family history. And I'm going to argue that today, family history is how we practice genetics in the clinic every single day. And most people would view family history as being important. So ask yourself this question. Do you think family history is important? Now, you may or may not, but I'm sure you all fill out those forms when you go into a new physician's office about your family history. And what's very, very clear, if you go in and look at some of the literature around family history, is that there's a whole big set of problems associated with family history. And so this paper, I think, is kind of interesting because it reports that men underreport significance. I don't think that should necessarily be a surprise. The other thing that they have discovered is that men have half as many relatives as women. And I'm sure if you stop and think about that for a moment, you'll realize that in many, many families, my family included, the woman in the relationship manages both sides of the family. Many men don't know who all their relatives are. So the lack of ability to remember, underreporting, and then the fact that we actually don't share this type of information amongst Americans very readily. I can't speak for other nationalities, but most of the time people don't have these discussions. We don't talk about what drugs we take. We don't talk about when do we take them. We don't talk about why do we take them. So I'm going to argue, at a minimum, genome sequencing is a quantitative family history that should improve every year. And how would we get to that? How would we look at that? Well, one place to go is to look at the genome-wide association studies. And this is an old slide. There's been many more traits that have been put on top of that. And those of you that are in the audience that are listening are going to be saying, yeah, but Howard, we all know that these have very minimal effects. In fact, they're such a small effect that they're really not meaningful. Again, this is on a population level, and I just want to remind you about a couple of points. And the one that I like most as an analogy is that salt also, epidemiologically, has no impact on blood pressure. But we know that in many cases for hypertension, salt does have a major impact. And I just want you to keep that in mind. So I would argue that if you have a SNP that's been shown to be associated with a disease and a reported family history, maybe we should think about that. And I think we're going to see more and more data around this. In fact, there's been a pretty nice publication that's come out of 23andMe that talks about how this information can be integrated with family history. The other part, here's a slide. I forgot that I put that in. This is the paper I'm talking about, and I think this is a good read. It goes in and it talks about where these different diseases are and where they can be used, and I'll talk some more about this as we go through the process. The other area, of course, that people talk about is using genetic information to help with pharmacogenomics. I, again, think this will be a big example down the road, and people will use it for this. Here's the example of clopidogrel, which is a commonly used drug. Its trade name is Plavix. And this drug is known that between 15 and 24% of the people that take this medication are poor metabolizers or are unable to metabolize this drug. And this information is not typically used clinically, even though it's recommended. And one of the big reasons around this, both for the family history SNP information, which people aren't sure about, and even in the cases for drugs, there's a major driver that prevents this. And the fact is that genetic testing for diagnosis and drugs is largely not used and often excluded from insurance plans. And that really starts to have an impact on how we think about practicing medicine. So I want you to think about that for a moment. I know these are the big issues. I'm going to walk you through some clinical cases about how we are being able to use this, and I will finish up in talking about how we are actually getting reimbursement for some of these types of tests. So I will come back to that as we go forward, but I want to now move you into how we got involved in using whole genome sequencing for a clinical test. We do offer it as a clinical test. We have both an adult clinic and a pediatric clinic that focuses on using whole genome sequencing as part of our diagnostic routine. So I'll talk about how we got there. So the next slide is a picture of Nick Volker. Nick Volker was 2 at the time. Some of you may have heard about this story. Some of you may not have. Nick was a healthy little 2-year-old boy, and then all of a sudden things started to go wrong. And I was not involved in the case early on, but I did get an email from Alan Mayer in July of 2009. Alan was the pediatric GI specialist who was working with Nick, and this is the actual email that he sent to me. that said, if you look down in the bottom paragraph down here, it says briefly, Nick, I can now tell you his name, was a 4-year-old boy with a very unusual malignant form of inflammatory bowel disease and profound delayed wound healing. He first presented with weight loss and perianal abscesses and was on a downward spiral for over 2 years. He was admitted in January. Mind you that this is July 27th with a life-threatening breakdown of his intestines, and they've been trying to keep him alive. So long story short, Alan asked, could we sequence his genome and figure out what was wrong with him? We, at the time, looked into this, and the best we could do would be whole exome sequencing, which was going to cost, just for the wet lab work, about $75,000. So we ended up figuring out how to do that, and we set out on this mission to be able to do that. And the first thing, of course, we ran into was, how do you manage the data? And in the case of an exome, it's going to be about 18,000 variants for an individual. And I'm just going to walk you through some of the activities that we went through. But this is what Nick looked like at 4. Doesn't look nearly as healthy as he did at 2. And the question was, what do we do with this? What's wrong with this child? So one of the things you have to be able to do then is to look at prioritization. So this is actually the data from Nick. How do you come down and look at this? So he had 16,124 variants. We went through a series of decisions, protein coding, non-synonymous, typical things that you would that you wouldn't necessarily do any different than we would do. And the part that was important is that since it's a rare disease, it needed to be absent from other reference genomes. We came down to 2 genes. One was GSTM1 transferase, and the other was XIAP. Long story short, we were able to exclude GSTM1 and focus on XIAP. If we have geneticists in the audience, you are aware that XIAP is X inhibitor of apoptosis. And this gene has been reported previously to play a role in a disease process that if this gene is damaged, patients will die if they get a infection from Epstein-Barr virus or from mono, and it just comes down to a lymphoproliferative disease. Going back and now having looked on it with time, we learned that in fact some of the case reports do list some GI problems, but that wasn't the major focus because they were battling the lymphoproliferative disease. So how do you move forward on this? Well, we move forward by looking at a bunch of different bits of evidence. We could show that it was in his mom, and since it's on the X chromosome, it kind of made sense for Nick. We went and looked into the evolutionary conservation. So if you look at that little box on the upper right-hand side with all the colors, there's a whole bunch of different species there, and you can see that here's the amino acid sequence. The amino acid C is conserved all the way down to fruit fly. And only Nick, still to this day, and his mother, as far as we know, carry this one amino acid change. Now, since she's got 2 X chromosomes, she's protected from this disease, so that was encouraging. And going in and taking a look at how does it fit into the gut disease, there was some nod signaling, and so there was some interest around that. But Dave Margolis, who is our bone marrow transplant specialist, said, you know, this is very encouraging, but I'm not going to do a clinical, I'm not going to make the clinical decision to do a bone marrow transplant, and I will not transplant him because there's no evidence that doing this transplant, even though you tell me about the nod signaling, is going to resolve the gut disease. You got to show me more. And so this was one of our, first cases of, well, how do we deal with this? There's nothing in the literature. Nick is not doing well. Do we pull the trigger on going after this or not? And that became a major issue as we went down the road. We brought in Jim Verbsky and Jack Rudis. And James then went after and looked at some of Nick's cells and went after the signaling, and was able to show that the signaling and apoptosis were clearly off in Nick's PBMCs, and was able to show that. And finally, Dave Margolis said that along with that information, and that was linked to nod signaling, and Nick wasn't doing very well, he would do the bone marrow transplant. And so what ended up happening then, 42 days later, after the transplant, Nick was doing well, and I will tell you that we just passed his 3rd year post-transplant, and he is resolved of all gut disease. So it clearly worked. That captured a lot of people's imaginations. It was a single case, and we've then built forward on how to build a project around a clinic to be able to do this. And so one of the first lessons that we learned was, do you have physicians who will practice medicine with the data you collect and analyze? And the corollary to that is, what data do they require to make a diagnosis? And one of the big challenges, if you're interested in implementing this, is really having a clinical team that's going to use the information. And it doesn't make a lot of sense to build out very, very sophisticated algorithms if the clinical team isn't going to use the data. And this is what, in our case, we believe is the biggest challenge of implementation. It's not the sequencing. It's not the data analysis. Yes, those are important, but it's how do you get the information and what information do you get to the physician? And I'll talk some more about that as we go through. The paper was published. We had a very difficult time publishing this. It was published in Genetics of Medicine, but because it was an N of 1 and it was pretty early in these days, we had a tough time on this. If you're having trouble sleeping at night, I guarantee you that you can fall asleep reading the paper. If you're interested in the more life events that happen around this, a couple of reporters got their hands on this early in the process. And followed the case, and they reported on 1 in a billion, which they ended up winning a Pulitzer Prize for, which it's a very interesting read in terms of what was the family going on, what did the family deal with, and I want to come back to that a couple more times later, because as we think about actionability, what families think makes a big difference. Okay, so where are we today? Well, this is just a little bit of a flow chart about all the different pieces that go into this for us, and we are, as I mentioned previously, have a full clinic. Here's all the steps that we go through in this. It is both CAP and CLIA certified. We have 3 clinical geneticists who focus in this space, and we have 2 genetic counselors, one that focuses on adult, that's Jenny Gertz, and the other that focuses on pediatrics, that's Reagan Veith. And these individuals then make up the team that's practicing the medicine. Everybody else is involved in supporting those programs, which we'll talk about. So one of the big issues, of course, is establishing causality. How do you establish causality? This is obviously a big challenge, and the issue that comes down to, unlike in a research project, we're trying to do the following. We need to determine whether this variant in this gene causes this disease in this case. And that's the fundamental piece around this. And can you develop the data around that your team believes and the clinicians believe, and that we're going to change how we practice medicine with? And that's a pretty big issue that most people are facing. To do this, we have to consider the annotations and the impact of the variants and the function of what is impacted. I'm going to talk a lot more about that. Obvious questions: is that variant real? Does the frequency of the variant match the expected frequency of the disease? Is the variant known or likely to be deleterious in function? Now keep in mind that there's, a few thousand people that have been sequenced. And most of these people we don't have disease information on. So, we're generating data and we're missing data to be able to analyze this. And obviously there's only a handful of genes that have been effectively studied to answer this question about the variant. Is the function impacted by the variant associated or associatable with the phenotype under analysis? And again, I take you back to the Nick story with XIAP. Once we did this, and once we did find some GI indications in some of the other case reports, and now we were able to resolve Nick's case, it's obvious that this was the right choice. But how many of your clinicians, and those of you that are clinicians that are listening, how many of you would have made the same choice? And I think that's a really big issue as we go down the road, and as different clinical societies try to gauge how do we manage this information that's going to all continue to happen. And so what's the level of evidence that's sufficient for clinical reporting? Obviously, having multiple publications makes sense. But what are you going to do if you have an N of 1, like we did in the case of Nick? So what we do is we break the evidence levels into a variant axis. We have one variant axis that's looking at the phenotype. So we need to look at this. So does the variant segregate as a causative phenotype in question for multiple families? Well, that's a pretty strong level of evidence. And as we go down this row here, you can see at each level there's less and less confidence that allele frequency or inheritance pattern is likely to cause the disease. So you have that piece of information. And those of you that are involved in undiagnosed diseases or involved in this process realize that the challenge sounds quite simple, the causative phenotype, but in many instances, it's very, very difficult to ascertain what the phenotype is. And so we've had to build out a series of tools. One that we use is called ClinMiner, which we now have to take the information out of the electronic medical records. In many cases, we're getting family members that are coming in that have massive volumes of data. And how do you get that data? And how do you go through that? Who reviews that? And how do you get that all in play? So we've had to spend a lot of time on that as well. And that's often not thought about when we think about genomics medicine. But the phenotype is fundamentally the driver. And I don't want anybody to think that I'm talking about genetics alone or genomics alone is really going to answer the questions. Our view is genomics is another lab value that needs to be put in context of the case that you're looking at. Now, as we start talking about secondary events or secondary indications or incidental findings, depending on what you want to call it, we'll talk about that a little bit more. So the next step then is to really look at the level of evidence. So you've got does that mutation seem to play a role? Is it causative in multiple families? And again, this level of information then helps you drive down the functional side. So you've got the mutational effects, and you've got its relationship, and then you've got the phenotypes and multiple family members. And what you're trying to do is integrate all of this information, and really what it comes down to is your software and your clinical team then needs to be able to come up with a prioritization scheme. And what are you going to do with this information as you go through? So the integration then allows us to place the variants into different reporting categories. And so we look at these different categories as follows. So the previously reported and a recognized cause of the disorder, so we would make a pathogenic report on that. Previously unreported and is a type which is expected to cause the disorder, we call that likely pathogenic. Previously unreported and is a type of a variant which may or may not be causative of the disorder, that becomes a variant of uncertain significance, or a VUS. We tend not to report out 4s and 5s. We tend to stop at the level 3, which is a variant of uncertain significance. And I'm going to offer to you that our biggest challenge right now is we have many different variants of uncertain significance, and so do others that are out there, and I'll talk a little bit more about that as we go forward. Now, since then, we've put together a whole clinical series of tests. Here's a bunch of the different phenotypes we've used. This has just been published in the last month in Science Translational Medicine that gives you a lot more details. But my only point here is to illustrate that we're using it across a wide range of different phenotypes. The most important part about this, to be clear on, that this is for undiagnosed diseases. This is really where we're focusing our energies on one side, and then we have another side where there's some executive medicine activities that are going on where we're trying to understand how do you practice this in a healthy population, which I'm not going to spend any time on today. So the findings, what have we learned so far around our pilot projects? Well, we've learned that we can obtain a diagnosis about 26% of the time. I'll tell you that, in fact, yesterday and the previous week, we had 2 more cases that have been resolved, and I'll talk about that. So we're now up to 8 cases that we've resolved out of these 23. We have 4 additional cases with the VUSs, and 2 of those cases are the ones that just got cleaned up. And what happened there is there were some publications that just came out that actually showed that those genes and those variants that we thought were causal actually were in multiple family members, so we could comfortably make that as a call. The current standard of care, for those of you that are not clinical geneticists or aren't in this space, is that for these undiagnosed diseases, that anecdotally we solve them somewhere between 5 and 10% of the time. So increasing that to 27 to 30%, which is what other groups are talking about, that's a big increase in terms of genetic diagnostics. Now, I'm going to argue that because these are rare variants, you simplify the search space by the fact that they're rare variants. And so I think that's going to be helpful. But the biggest problem we have is there's not enough data to help us reduce the number of variants of uncertain significance. And so I think that's a major area in the field, and I'll talk just a little bit more about that down the road. So the lesson that we've learned around this is that the data analysis and knowledge must meet the clinician needs. They don't make decisions the same way, and how you put together your clinical program around this is critical. And this is absolutely essential as you're building out your decision trees and your software to help the physicians use this information. Giving them 18,000 variants, or in the case of whole genome sequencing, which all but Nick has been done with whole genome sequencing. So Nick was 16,000 variants. Most of our other cases that we're working with now are about 4 million variants. So handing a physician team 4 million variants is just not helpful. You really have to focus your energies around that. And I'm now going to move into talking about some of the relations that you have to think about, or relationships to the data you have to think about, in addition to providing the clinicians information. So problem number 2, what data will be returned and who owns the data? And this is an absolutely essential piece to this, because it shapes not only what you're going to give to the physicians, but what type of information the physicians are going to use. And I'll give a very simple example. If the physicians are only going to return data related to the phenotype, you don't need a very complicated pipeline. If you're not going to follow up, you don't need a very sophisticated data storage. If you're just going to do this one time, you're going to tell them the data, this is related to your phenotype, and you're done. Again, what you have to save and what you have to do is very different than if you're going to do follow-up. And also what we've learned is that, are you going to require other family members to be sequenced? There are some people out there that believe we should only be doing this in trios. We do know from experience that if we had done this, some of our families would not have signed up to do this. We do require, in the case of children, that the parents provide DNA to check where the alleles come from and to look for a de novo. That's the only thing we require. We do not require the parents to be sequenced. As I said, we do also know that if you require that, in some cases people will, they view that as being research. And while we may all think that research is fantastic, many of these families are sick and tired of not having their case resolved, and view research then as another way of not trying to help their family member. So the ACMG just came out recently with some recommendations for reporting on incidental findings, or what I'll refer to as secondary findings in clinical exome and genome sequencing. They come out with some pretty strong statements about what types of data need to come back. I am happy with some parts of this. I was quite concerned that there was going to be some resistance around incidental findings or secondary findings in totality. I think the challenge here is that making sure that these data have to go back to the physician, and putting the pressure on the physician about that relationship with the patient, is quite challenging. And what do I mean by that? If the patient says they don't want to know about BRCA1, and you insist on getting the BRCA1 data back, what does the physician do? Does the physician choose not to do that? And so I think we could have a long discussion, and maybe in some of the questions that will come up, there's a lot of discussion around this. And I think it's premature to make categorical decisions about how we must do this. I think as a field we have to be careful and really do some experiments around this. And so we've done that. We've been looking at a categorical model of choice. Our ethicists find this to be an acceptable strategy. And in Wisconsin, if it's diagnostic or treatable childhood, and we're talking about a pediatric case, we are mandated by state law to inform the parents of that information. Optional disclosures, nothing else. Non-actionable childhood, actionable adult, and the one that really makes people nervous around this is non-actionable adult, meaning something like Alzheimer's, something that's not treatable. Is it the parent's right to get that information when the child may not want to know that themselves when they turn 18? And so that, I would agree, is a major area of consideration. And so what have our cases come up with? Here's the exact data from 13 cases where we looked at this with. Only one family of the 13 chose not to go forward with whole genome sequencing. And of the 12 that then went forward, only one chose to take nothing else. The interesting one is over 50% chose to have all data that comes back. So we asked ourselves, well, is that a good thing or is that a bad thing? So David Bick and some other team members have been going out and doing surveys when they give lectures to us. the colleagues that are working in this space. And this paper was recently published, and the argument would be that if we all believe that adult actionable shouldn't be displayed, then when we're asked about our children, we should all say, no, we don't want that information. So what did we, the learned, decide? Well, this paper I think is quite interesting, and if you were to read down through this, 44% wanted to know adult non-actionable in themselves, and 31% wanted to know this about their children. That's not too far off from about the 50% that we saw before. And I think this just simply illustrates that we as individuals and as parents believe we have the right to have this information. And I think there's some very powerful information around that. And I'm going to come back to that again when we talk about personal utility as part of what's actionable with this data set, and something to keep in mind as you go forward. So again, I think you can see, if you're going to give back all data, or you're only going to give back what's required by law, it makes a very big difference in your pipeline and your clinical decisions and what types of things you have to do and how you want to build it out. And again, there's really no rules exactly in how we need to do that. So the lesson we learned here was that data return rules impact the data analysis, the storage, as do the follow-up plans. And I'm going to argue that that this is really a big deal, and when you're talking about building out your infrastructure. Having physicians is obviously essential. If you don't have physicians that want to do this, you don't really need to worry about the rest of it. But if you do, this is really where you start making decisions about what's going to impact your cost to implement this. So problem number 3 is what's actionable. I've been using that word back and forth. I think we all would have an opinion about this, and in fact, I've been quite interested in finding out what people view actionable to mean. And what I get asked often is, well, from our point of view, being medical professionals, actionable means is it something we can do about it? That, why test if we can't do anything about it? There's a fair number of people that don't buy the argument that family history is going to be helpful, and this is really going to change the care. So why should we spend this kind of money when it's really not going to make a difference? And I'm going to argue, and I'm intentionally being provocative as a way to help everybody think about this. So I'm going to argue that we do this clinically every day. We test things that we can't do anything about every single day. And I'm going to just give some examples that I think illustrate the point. Only 25% of our patients with high blood pressure actually have controlled blood pressure. And in fact, we know very well that those with high blood pressure still are dying earlier of end organ damage, even though we claim we can treat it. Cancer, diabetes, and obesity are all areas where we test, we make diagnoses, and we have, in often cases, minimal levels of success of being able to treat. Yet we don't ask the question, is it worth testing and worth not testing? And I think it's something to keep in mind. And again, this is very, very personal, at least from a practitioner's point of view. Should we be doing this? Should we not be doing it if we can't change outcome? So here's another case. This is Lillian. So Lillian was a 40-week-old ex-preemie. Had a lot of pregnancy complications. She had IUGR. She had a whole host of issues that came out around her disease process. She was not treated here. She wasn't born here. Following an emergent C-section, there's complex phenotypes that you can see here, presented with an infection, increased severity of diarrhea, and was in the NICU for 3 months. And they went through all kinds of different events, tried to figure out what was wrong, and long story short, they were not able to figure this out. So mind you, we saw her at 4 years. So this family had been on one of these diagnostic odysseys where they'd gone from clinician to clinician, hospital to hospital, trying to find an answer around this. After the extensive evaluations in GI, we ended up doing whole genome sequencing, and what we found was a candidate variants in 3 different genes. We were able to exclude a bunch of these, and we were left with SCIV2L. It had an ultra-rare het that showed no frequency data that was out there, and there was another SNV, or you can think of that as a SNP. A SNP and an SNV are the same. In case it's a single nucleotide variant as opposed to polymorphism, we prefer that term, causing the formation of premature stop. It was, as before, highly conserved, so it seemed logical. And then the other was a canonical novel splice variant that looked like it caused a frame shift downstream. So it looked like we had basically found this. Now the challenge with this is this the gene or isn't it the gene? So how would you take a look at that? Well, of course, you go to the literature. And what we found was 13 papers had nothing to do with this phenotype. In fact, looking at this, you wouldn't even think that it was involved in any of these phenotypes at all. But what was interesting is that if you now go take a look at the molecular biology around this, SCIV2L interacts with the TTC37 gene. Now, this gene has actually been implemented with a hepatoenteric syndrome 1. And so we had actually looked at this gene as a potential causal of her phenotypes, and we had excluded it based on the testing. Well, now we have an interesting point of view. Well, now we have a binding site or a binding partner to this gene that has been implicated in something like this. So would you make a diagnosis that SCIV2L is actually causal? Would you make that a pathogenic? Here we've got some evidence. Now, in our case, we said no. This is a variant of uncertain significance because there's no evidence that this is actually causing the disease. So this was a variant of uncertain significance for months. It was the top of our list, but it just sat there. And it was categorized as a variant. It wasn't put out there as a likely pathogenic. It was a variant of uncertain significance, and we had no other case. Yet alone, what happened then is a paper came out, which we're very happy about months later, that this group had actually looked at 6 different individuals. And indeed, it is a variant that is actually causing this, and they proved that. So suddenly, the only thing that changed, and I say only in quotes, it's a big deal, was that now this paper came out, and it was able to prove the point that this was the diagnosis. And so we gave the parents a diagnosis. It has helped us think about how we manage the care of this child. It has helped us do it better, but there is no cure for this. This is not a Nick Volker story where we're able to come in and figure out how to resolve this and solve some problems. And in fact, it's introduced us and the family to have to think about some other potential places to worry about downstream. So it has modified care, but it hasn't changed the child's life. And so some people would argue, well, that's a good deal. Some people would say, well, have you really changed care? And so we think we have. And so there's no cure, as I just mentioned, and additional family members have been tested. And of course, for this family, since they're a young couple, it could have potential impact for them downstream when they're thinking about having other children. So these are ways to think about this. All right, so what I want to do is have you listen to the family. So this was obviously, as you listen to this, was put on as a marketing campaign for raising philanthropy, so I apologize for that in advance. But I want you as the audience to listen to what the parents say about the meaning of the sequencing to them. So if you'll play that, please, Don.
Our whole life, since Lillian's been born, has been kind of like a roller coaster ride. She was born at 30 weeks, 1 pound, 4 ounces, 11 3/4 inches long. She was ill several times in the NICU. She was tested for Shwachman-Diamond syndrome. She's been tested for cystic fibrosis. She slowly stopped eating anything by mouth, and we just did tube feedings. She's had 3 colonoscopy endoscopies. I can't even remember all the different things she's been tested for. They had been to many institutions. They had been seen by many doctors. They'd been seen by geneticists. They'd been seen by all sorts of other subspecialists.
She had been through so many different tests, and with them all coming back negative, I think that at times that they wondered if they ever would find a diagnosis.
She became sick in fall of 2010. I remember looking at her one day and feeling like she was dying in front of me. She had gone from 24 pounds to 18, and they couldn't figure out what was wrong. So that's when we came to Children's.
You know, the whole concept of the whole genome sequencing was new to us.
Genome sequencing is a process where we examine or we look at all of the genetic material, all of the DNA, all of the instructions that a person has.
Its purpose is to help us diagnose children when we've not been able to find a diagnosis before. Many physicians suspected she might have a genetic condition.
She really fit many of the criteria of a patient who had an unexplained disorder and for which there may be potential treatment strategies.
Genomics is almost our fulcrum, if you will, to the reach out to the many specialties that our genetics program supports.
One of the remarkable aspects of whole genome sequencing is really the range of patients that we've been able to help. Patients with intestinal problems, patients with immune problems, patients with neurologic problems, patients with cancer. The list goes on and on.
If they could find something that would tell us why Lillian is who she is and why she has the problems that she has, that was our number one thing.
Anything to give her a better life. When we finally found the molecular diagnosis for Lillian through her genomic sequencing, we were just ecstatic. I couldn't wait to pick up the phone. I couldn't wait to call them.
She's the first child that's been diagnosed with this in the United States. She's unique. The diagnosis is unique. We have an answer. It gives something for the specialist to continue to look into to see what they can do to better help us.
Lillian has made tremendous progress over the years. Every time I see her, she's doing better and better.
By doing the whole genome sequencing, we don't have to keep searching. If it weren't for Children's Hospital, we're not sure if Lillian would still be here with us today.
Okay, so several points. Obviously, the family was happy with this, with the diagnosis, and I think the two things that were very important that the parents said was they have a diagnosis, Dad said that, and Mom said we don't have to keep searching. And I think as we start looking at personal utility, there is tremendous value around for this family, and I think that these types of situations and people wanting to know is going to continue to drive into direct-to-consumer genetic testing if we don't pay attention to this. And I think this is really important discussions that we need to have. I'm not saying that we shouldn't worry about how do we make diagnoses. I'm not saying that we should turn this into the wild, wild west. But what I am saying is that we have to look at utility as being more than just what we can do to help these patients. There is something about having a diagnosis, as you all know, that's meaningful to a family member. Okay. So if we now continue thinking about this, how you define actionability and what's actionable and determine when to deploy clinically is going to have huge effects on how you build your program. If you're going to really not worry about anything that isn't druggable, that you can't treat, then you don't really have to spend a lot of time on this. If you're going to really try to go after making a new diagnosis, then you need to think about this very carefully. And in fact, you know, we hope you do want to do that because collectively, as I'll talk about in a few moments, we're going to have to work together to solve these variants of uncertain significance. Now, what I also hear over and over and over again is no insurance company will pay for this anytime soon. This is a letter, I can't tell you the name of the insurance company, that was written to David Dimmick back in February 23rd of 2011 that says, as we have discussed, in situations where you determine that on average the cost of routine testing will exceed the contract price of whole genome sequencing, we will authorize whole genome sequencing as a first-line clinical test. So I'm a big believer in testing hypotheses, so we asked the question, and I'll ask you, of the 12 cases where we were following this on, how many do you think we had pre-authorization on? So just think about that for a moment. Typically, we get people saying 0, and we get people saying 12, which was great because it turns out that 6 out of 12 got pre-authorizations. But I'm going to tell you that it's not necessarily a slam dunk. We basically are going to the insurance companies, and we're basically arguing that we'll start with a gene test, we'll go to a gene panel test, we'll go to a whole exome, and then when we don't find it, the last reflexive test we're going to do is whole genome sequencing. Or you can do whole genome sequencing. And in some cases, they've really decided that they don't want to roll the dice. It's cheaper to actually do the whole genome sequencing in advance. Okay, so I want to summarize and finish with a couple key points here around what we're doing. So we have a clinically certified laboratory and analysis pipeline for whole genome sequencing, whole exome sequencing, gene panels, and we do follow-up clinical Sanger sequencing as a validation. So we use next generation for the sequencing, and then we do validation with Sanger sequencing. Because of this, we actually have the ability to test any variant or any combination of variants clinically. We had to figure out how do you put together a clinical test that any variant that pops up we can actually use. And that's turned out to be a bit of an advantage for us because we have people contact us and say, hey, could you check this variant in a clinical setting? And the answer is we can do that. So that's been helpful for us to think through that. As I mentioned, we've developed a clinic for adults and children and are practicing this medicine with whole genome sequencing. And the reason we call this personalized medicine is because the decisions the families and the patients have to make is incredibly personal. And if you've been involved in this from the counseling side, then you also know that this is a big deal for these families. What incidental information do you want back? And we can think about this esoterically, but these are real questions that people have to make. And that's why we consider this to be personal, because of the nature of the decisions and the impact on the individuals and the families. We are making a diagnosis about 25% of the time. It's just bumped up. As I told you, we've solved 2 more cases this week. But between 20 to 30% is what most undiagnosed disease programs are talking about resolving right now. And if you've heard other people talk about this, they're going to come up with many of the same things that I've told you, which is these variants of uncertain significance is the challenge. We do believe, as I've mentioned to you, that actionable does include personal utility. We've had some very interesting results with our families and their patients. In another case where the child died and the family decided not to do a transplant because the child was going to die independent of the transplant was just going to delay the death. The family, while not happy with the diagnosis and outcome, had some relief that it was not their fault. They hadn't done anything wrong. Again, very much in a personal utility category. I'm going to argue. We have a lot of people that debate whether you should do it or not do it. I'm going to say do 20 cases at your institution. I will be shocked if you don't find what we found, which is it does have incredible utility. We have also had an increase in our physicians' interest in this. I'm no longer having to go around on campus trying to convince people to use this. We're now worrying about how do we implement this across different clinical lines, because now the clinician-to-clinician conversations are, have you tried whole genome sequencing? And I think that's been a big change for us. I think you can impact medicine today. I urge you, if you're not in a point where you can think about doing whole genome sequencing, to get people starting to think about it. Pick a gene or a panel, but just get started on it. This is coming incredibly quick. Look, we're not, an institution you would think about doing this. We're the Medical College of Wisconsin. Probably many of you have never heard of us. If we can do this, this is going to happen relatively quickly. And the other part that I think is important, why are we really pushing this? You having your patient sequenced and we all sharing data is going to be what's going to be absolutely important in us figuring this out. So here's an example of a case. You can see this case on a Nova show called About Understanding Your Genome, where we were on Nova and we successfully failed. I'm going to say that again. Successfully failed to make a diagnosis on our case 48, which down in the lower right-hand corner, we didn't make a diagnosis. But in working with the NIH through their undiagnosed disease program, they thought they had a couple of other cases that had a similar phenotype. And long story short, they've resolved this case. We didn't resolve it up front. But this is a success. The fact that we had done the sequencing and they had been involved in the sequencing themselves, they wanted to do it themselves, which is absolutely okay, and integrating the data, they actually found it. And they were able to figure this out. It's not the same variant. But if you think about it, once you know the gene is causally related, now you can go back in. So we've got to be thinking about this as a community. How do we share this data? And because of that, we've now had a diagnosis for case 48. It is probably not going to change our ability to treat case 48, but the family, again, has consolation in knowing what the diagnosis is and knowing what the disease process is. And the functional studies are underway, both at our place and at NIH. So why are we doing this now? Here's the last case I want to present to you. This is Avery. She initially presented with poor feeding and failure to thrive, but met early developmental milestones, subsequently began to fail. Many tests were performed. Nothing abnormal was identified. After 3.5 years, it was determined that, in fact, she had a treatable disease. And she's got some great outcomes now, in that the neurological degeneration has stopped. But what happened before, we can't change. And nobody made a mistake on this. It was just the clinical presentation in her case didn't suggest that it was this. There's many different gene tests, many metabolic tests that were done, and it didn't show up. Had we done whole genome sequencing on her, we would have found the variant and we would have known what was wrong. So the reason we do this right now on our institution and what we're focusing on is we're really trying to avoid these types of situations and really trying to prevent that. So that's where we are. I want to close before I start taking questions to tell you that if you want to obtain your CMEs, you need to go to this site and pick up that information and go there and go through. I think you have to take some quiz along that. So with that, I'm going to close and take questions. I see we have Q&As that are on there. I will open those up and start answering questions around that. I'll get through as many as I can. So let me open that here. Okay, so I'm going to take a couple of these questions. I will tell you what the questions are, and then I will try to answer the question the best I can. So since this was the beginning, how did you get insurance companies to pay for these tests? I'm going to be completely honest with you. There was, I don't know how old the age group is that are around there, but what we did is that we basically showed them that you could pay us now or you could pay us later, the old Midas commercial, that we basically told them that we were going to nickel and dime them to death. And we had several cases where we put it in and it was rejected and we were told, you know, please appeal. And I'll tell you, I've met with several chief medical officers for the insurance companies. They're paying attention to this. Everybody's just trying to get their head around this. And so it's not a slam dunk. You just submit this and then it happens. It really is something you have to work through. There's some other questions that are related around this. In terms of costs, I'm happy to share with you the costs. We are doing whole genome sequencing, the entire counseling, sequencing, analysis, data return, incidentals for $17,500. It's a 90-day turnaround. We are doing whole exomes for $48.75. Again, that's I think that's a 90-day turnaround as well. There's a question related to who would pay for Canadians who would come for care at our institution. I don't know what the answer to that is. If there's a specific case that you're interested in, please feel free to email me, jacob@mcw.edu, and we can put you in touch with that. In some cases, I'll be completely honest with you, families have decided that even if the insurance wasn't going to pay for it, they've decided to pay for it themselves. So there are solutions around that. It's not something that I would prefer, but that's kind of where we are on that bit. Let's see. A couple other people are agreeing with the generalized strategy and think it's got some great potential. Let's see, other questions. How will the diagnosis be impacted by the Affordable Care Act? We have no idea. Again, I think this is something everybody's trying to figure out as we go through. You know, I'm not sure there's going to be a solution to that anytime soon. I think we still have to keep pushing on it to help make it be a reality. Another question, in terms of success rates for making a diagnosis, is there a significant difference between whole genome sequencing and whole exome sequencing? That's a great question. The answer is that we have found in the 8 cases now that have been solved, I believe it's 2 out of the 8, we would not have identified with whole exome sequencing. Now, one of the challenges with whole exome sequencing is coverage. We actually get better coverage of genes when we do whole genome sequencing. So it's a little bit of a hard question to answer. So we obviously get the same data that you get with whole genome sequencing as you do with whole exome sequencing. If you're not familiar with whole exome sequencing, there's a pull-down step. And that pull-down step has a bias that's associated with it. So the advantage of whole genome sequencing is that you actually can get some better coverage and some key genes that are missing in the whole exome. So it's a little bit of a chicken versus an egg question. The other part that we do in each case, whether we do the whole exome sequencing or do the whole genome sequencing, is Liz Worthy, who's built our software, her team's built something called GapMine. And GapMine actually goes through and categorizes which genes are missing, which exons and which bits. So we can then go in, and if the clinician team thinks that that's a major fault, then we can go back in and fix that sequencing. So to us, we still think that whole genome sequencing offers a reflex for groups that that can get a whole exome sequence. If it doesn't work, then perhaps whole genome sequencing is another way to get the insurance companies to think about this. Any other questions out there? We're sitting at 5 minutes left. I'll wait another minute or so if there's any other questions. Otherwise, for those of you that are on, thank you so much for your participation. And like I said, oh, there's another one that's come in. So, yeah, so was there validation after the treatment with another whole genome sequence to ensure that was the SNV and causing the disease? What we do in all cases, we take the next generation sequencing, and before we report it out, we go and we validate that variant in our Sanger sequencing protocol. That's part of our clinical delivery. So part of our cost then includes validation or verification, maybe would be the right term, of the variant. So we do that. We do see about a 6% difference between variants that we think were called in the next generation sequencing versus the Sanger. So those are validated in that way. I'm not sure if that's what you mean. In some of the other cases, we also then are going in and we're trying to do some studies around those. And we're, again, looking to find other family members that are out there that have had their genome sequenced to help make the decision. I believe the guidelines right now, although the ACMG is working on this, is that you need to have 3 unrelated examples of a polymorphism that's been associated with the disease. There's some talk of moving that up a little bit higher. And so you need some of the biology also assigned to that. And so we have some strengths in some specific areas, and in those cases, our immunology clinic, as an example, we'll go in and do some gene editing and cells and try to see if we can prove that particular variant. The key to that, though, is I'll only do those follow-on studies if the clinical team is willing to make a diagnosis based on that change. If the clinical team is not willing to make a diagnosis with a follow-on study, then we declare it as a VUS and hope that somebody will publish something on that sooner than later. Any other questions? This question, the next one just came in, is what sequencing platform are we using? We have basically, we have quite a few different machines. We have a 454, we have Illuminas, we have a PacBio, we have a MySeq, and what we're doing, the bulk of the sequencing on is the Illumina HiSeq. it's got its good points and its bad points around it. I think all of the short read technologies have some equivalency to them. I would like us to get away from short read technology and get to a point where we can do de novo assembly with longer reads, which is why we're looking at the PacBio as a potential solution. I know Illumina is looking at, they've got some new technologies that are out for longer read lengths. So for me, it's really read length and quality and the price point. And so for us, Illumina has just been a better solution. I'm not saying it's the only solution. I'm just saying that that's the solution we've chosen. Let's see. Yeah, so the question is around how do we get the insurance companies' confidence in the technology? We need to look at sequencing before and after. We do have a couple of studies that are underway to be able to look at some of those questions about is it really, is it better? And that's just going to take some time. And so there are quite a few groups that are out there that are waiting to see those data. I think that's going to take a fair bit of time to convince people. I'm hoping that what will happen is that we'll get to a point where the insurance companies will start to realize, and I think cancer is going to be the first place, where they're going to realize that sequencing tumors and looking at sequencing of RNA and coming up with expression profiles for that is a way to help determine which drugs the patient should get, since these are incredibly expensive drugs. I'm thinking that might help drive that as well. I think it's just the big concern, as near as I can tell, is people are worried about what's going to happen is with the incidental findings that we're just going to drive up healthcare costs because there's going to be all the follow-on studies, all the follow-on clinical characterizations that are going to go on. And so I think we're just going to have to prove the point. The analogy I like, some of you may not like it, is the MRI. When that first came out, there's whole body screening, there's all this other stuff. And with time, both the insurers and the practitioners and the patients, actually all three, have figured out what's a better use of MRI. And to some extent, I'm afraid we're probably going to have to do some of that with whole genome sequencing. Okay, we are at time, so I'm going to close. I want to thank you for your attention. Thank you for the questions, and hope to see you out there sequencing clinically soon. Thank you very much.